Assalamu alaikum. Today we will discuss about the bioprocessing, various types of the bioprocessing processor designs, including the batch, fat, and continuous fermentation types, and their advantages, disadvantages, and their application in the various field. But first of all, we must know that what is meant by the bioprocess. Bio, you all know that's related to the life. But here will consider that if we are interested uh, to get some useful products from the microorganism, then through what process or through what types of these steps or through the series of these steps we can get these products from the microorganism. That's why the whole this technique is called the bioprocessing or the basically this is a technique in which you can use the different types of the microorganism, and in this way we know that microorganisms naturally have the potential and they are able to produce different types of the enzymes, antibiotics, and so many chemicals, and we can exploit their natural potential for our benefits. But through what process, through what types of the steps we have to take, and what type of the technology is needed uh, to obtain these products from the microorganisms. That's why we need the help of the other fields like the engineering and other things, and various types of the instrument needed, various types of the chemicals, materials, and methods are needed to complete that process. Basically, this process, there are the two main steps, upstreaming and the downstreaming in this whole bioprocessing technology. Look at this diagram. Here you can see the pure culture, and before that we have optimized the different culture conditions, sterilization, and various steps of the characterization of the microorganism. Then after getting the pure culture and through optimization, and then there is a cell culture. And now you can look at this. This is a pilot production. Pilot production means before going to the large scale production for the industrial purposes, small scale production is obtained under the controlled conditions. For example, 10 liter, 30 liter, 40 liter, 50 liter, 500 liter. But while when we talk about the industrial scale, it is a huge production. So before to step in the industrial scale production, first of all, the pilot product is needed. And then this is the at large scale production. You can say at industrial scale production. And now, what type of industry we are going to consider for the large scale production, and what type of the products we are interested to get or to downstream. This whole side is showing the upstream process, and while this is the downstream product process, how we can get our products. And this is a simple diagram. You can get an idea how control conditions matters a lot, how the nutrients are added, and how other parameters, pH, temperature, oxygen, and substrate level can be optimized to get to a large scale production. These all three steps are different in terms of the advantages, disadvantages, and their application. And then, as I earlier told you, that in the case of the batch fermentation, all nutrients, microorganisms, and other things managed at the same time, the start of the process, and it is a closed system. It has advantages and disadvantages. In remaining slide, we will discuss what type of the advantages and disadvantages and application every method has. Look at this diagram. There is a biofermenter, or you can say the bioreactors, and how is in the laboratory or industry it is connected to the various types of the system. In the fed batch fermentation, everything is optimized in which and the nutrients and other things are introduced gradually with the best of time. And in this way, process get continuous, and we can get our byproducts. It has also advantages and disadvantages. Here are the mentioned the key advantages, but we will discuss in next slides. In the continuous fermentation type, it is in this type, which is a continuously means non-stoppable. It's just like a flowing river. Just uh, nutrients are added continuously, and byproducts are released, and everything is managed and optimized. And it is very it has many advantages, but as well as disadvantages. And this method is ideal for producing the large volume of the low-cost products such as the ethanol and bulk other chemicals, suitable for the large-scale production. Uh, for example, you can say the production of the ethanol and bulk type of the chemicals if we want to produce them and at industrial scale, but they have the low-cost products. In this way, you can compare these three types of the bioprocess method: batch fermentation, fed batch fermentation, and continuous fermentation, and their advantages, disadvantages, and their ideal application. And then I think you can have an idea about what is meant by the bioprocess, what is meant by the bioprocessing technology, and what type of the bioprocessor design can be used. But it depends how we can select the best method for the industrial scale production. It depends on the nature of the chemicals and nature of the interest, nature of the product, as well as the upstream process matters a lot. How we can get the pure culture, how we can sterilize them, and how we can run our pilot experiment, and then we can decide towards the large scale production. It's not an easy process. It's very challenging, and it takes too much technology and understanding, and approaches matters a lot, and as well as the industrial demands. This way we can discuss what is meant by the bioprocessing and how we can use the various types of the microorganisms, because microorganisms are very small in sizes and they are easily culturable, and downstreaming process is very easy as compared with the plants and animal-based products. Mostly when we talk about their microbial enzymes, uh, no doubt plants and animals can synthesize various types of the enzyme, but mostly researchers and the scientists and the industrial scale, microbial enzymes are preferred at industrial scale because they are environmentally friendly, and they are easily culturable, and they can be easily separated out from the microorganisms, and they can easily characterize and easily optimized, and uh, cheaply chemicals are enough to get their enzymes. They are the economical process when we consider the microorganisms for the industrial product production, because microorganisms are independent. Uh, they do depends on the various seasons and the places. They can rapidly grow, independently grow. That's why it's very easy to work with microorganism in the laboratory as well as on the large scale production. But there should be a proper optimization of the various culture conditions, pH, temperature, various types of the substrates, and the other requirements of the microorganism. If we are really interested to get ideal products, I think this is enough for to have a basic idea about the bioprocessing technology. It is a technology. It's a very ideal technology or scientific methods, but we should follow the proper processes and the proper actions, or the proper series of the actions, or series of the actions are needed for to complete the industrial scale production. Okay, bye. Allah Hafiz.